John, thank you for the introduction to talk here today. Of course, I am delighted to have an hour to talk to people that I'm sure is very interested in this topic, on a topic that I find extremely interesting. And of course, the gut-brain connection is the one field where in the last 5 or 10 years, there has been huge breakthroughs and great advances made in the field that really fundamentally changed how we looked at this very important circuit. And what I hope to do today is to take you through some of the aspects of the consideration of what drives us as human beings to eat and why do we eat. Then looking at some of the great experiments that was done in the last 5 years, and to sort of wrap it up, looking at future perspectives and some new technologies, especially epigenetics and the inheritance of obesity, to give you sort of an overall flavor of where the field is at this point in time. So if I can move to my first slide. Here we go. So one of the major things that set us apart from any other organism on this planet is our very unique eating behavior. So as human beings, as sitting down for meals with other humans is a fundamental aspect of human behavior. And this is not something that is readily practiced by any other mammals on Earth. We spend a lot of money and a lot of time to process food outside of our bodies, compared to sheep, for example. Food is prepared and consumed at a particular time of day, at a designated place. We almost always eat in groups. Eating alone is seen as being slightly, sad or lonely. And looking at my breakfast table, we eat mostly peacefully, which again is a hallmark of human eating behavior. So for humans, eating is both social and nutritional. And humans have, there's this general idea that we've evolved to respond and adapt to food shortages, because typically we've been exposed to the cyclic availability of food during summers and winters. And this is sort of the genomic landscape that we are embedded in. And this general idea of a thrifty genotype and phenotype that was mostly selected by food insecurity and hunger in our past sort of evolutionary biology. So, of course, as humans, we eat for very different reasons. This little guy's obviously just hugely determined to eat. You can see the expression on his face. And I always joke with the students, I say, this is me at the end of Trinity term. So a lot of patients or obese patients often say they eat in response to emotions. Eating calms them down. Eating makes them feel better. And of course, we eat because it tastes good. Our brains reward us very strongly for the consumption of fatty, sweet food. We eat for social factors. And I think it's really important to pause on this for a little bit, because all of the major events in our lives are celebrated with food. Births, christenings, weddings, all of the religious festivals in this world is associated by consuming specific food. We often get together in family groups and cook these food before this particular festival. So it forms a fundamental aspect of how we socialize and the family bonds we create. And also our identity, our cultural and religious identities are very strongly wrapped up in food. And of course, our daily intake of food varies greatly. And one aspect that I actually forgot here, of course, is also the social status and demonstration of social status with food. So at colleges, we also take great pleasure in comparing each other's chefs, bragging about who has the best chef or the best wine cellar. We often would try and impress friends or rivals with expensive wine or good food. So that's again, very, very fundamental part of signaling social status to the prices of the food that we can afford to eat. And usually our daily intake of food can vary greatly. And of course, the timing of food consumption in December, for example, is very different from our food consumption in January. But in general, the cumulative energy intake is usually matched with energy expenditure with great precision over time. So this is the standard normal physiological conditions that we look at. And if you look at any sort of experimental animal groups, rarely overeats when you left them to free feed in their cages. And energy homeostasis promotes stability in the amount of body energy that is stored from fat, And you usually would have one winter's worth of adipose tissue that hang around. That was sort of the general idea of the borders within which humans will pick up and lose weight. So it's not usual for humans to accumulate a large amount of adipose tissue over a lifetime. And of course, there's been several books written on this topic. And looking at the social side of eating, of course, this is one of my favorite quotes that I often give to my FHS students. So this is written by Judith Martin, and she's a columnist, which I think very aptly describe here the relationship that we have with food, even as far as courting behavior is concerned. But she says, of course, under no circumstances can food be omitted from the way that we interact with those that we love. And it's very entertaining reading. If you're ever stuck on a train or a plane, I can highly recommend getting one of her books. But Power and Shulkin had written a very compelling book. This is about now 10 years ago, where they looked at the evolution of obesity. And this was round about the time when the scientific community was really starting to take notice of this increase of obesity prevalence in our general population. And what they pointed out is that the metabolic changes in mammals fattening up before winter, it's very similar to the changes seen in type 2 diabetes. So the genes and the evolution is there to accumulate a large amount of body fat. We certainly set up to do that. And of course, our genome sort of has been interacting with a dramatically changed modern food environment. And this is specifically relevant in the last 100 years. So especially after the Second World War, there was a very clear shift in the Western diet that happened more or less in the UK, Europe, and the United States at the same time. And obesity is generally classified as a failure to maintain weight homeostasis. So so generally, it's often this disruption in homeostasis is often sort of attributed to the modern diet. But what exactly this modern diet is, there's a lot of differences of people defining what the modern diet is. But the general idea is over-processed food that lack fiber and that contains a large amount of fat and sugar. And of course, John Judkin in 1972 was the first scientist to really raise this issue that we may have with adding too much sugar to our food. But of course, it's much more complicated. You know, the physical exertion associated with gathering food is no longer a major factor. I think, well, anybody that goes to Sainsbury's on a Saturday may feel like entering into an episode of Gladiator, but still that's very far removed from your standard harvesting and planting of food. And of course, we have easy access to high-calorie food all the time. We humans have a strong hedonistic drive to eat. We are able to refrain from eating when we're hungry most of the time if we're on a diet. And we can eat when we are not hungry. Again, this is quite human behavior. And if all of us would be fasted for 3 hours and given free access to food, every single one of us will eat for a different period of time. The amount of food we eat will be different. The type of food we choose to eat will be different between all of us. And how quickly we feel hungry again will be different. And I think it's this complex combination of how much, how long, what, and how frequently we eat that makes it very difficult to pinpoint the centers to target a specific center for treatment of obesity. And of course, we often overfeed when given the chance, because that's our way of fattening up for winter. So the regulation of feeding is complex, and these neural networks are spread over both the primitive and the higher order functions of the brain. So again, finding a magic target that you can single out for the treatment of obesity has proved really elusive over time. So the main alarm bell was rung by Hiller et al. In this paper that was published in Science in 2003. And this showed this dramatic shift. This was a National Health Examination Survey that showed this dramatic shift in the US population's BMI, so body mass index. If you look at this black curve, 6 years later, if you look at this red curve. And then the projected curve, of course, was the blue for 2008. And of course, that curve was actually even more shifted to the right. But what Hiller pointed out was not that what was alarming wasn't the fact that you had a sort of a general shift of the left to the right, but what we were really looking at was the emergence of a population that was particularly susceptible to this modern diet. This population of humans would overeat when they can and was terrifically effective at storing this additional energy as fat. And this emerging population was the one that was causing real alarm. So, of course, for this audience, I don't need to, I'm preaching to the choir. I think all of you are very aware of the obesity epidemic at the moment. But to highlight these statistics as the number of school children that are currently overweight and the number of these children that are under the age of 5. And of course, children born to overweight mothers are threefold as likely to become overweight themselves, and 77% of obese children become obese adults. And this statistics would clearly indicate that if obesity intervention should be aimed at families and children, this is where we will have the most effective impact. But of course, the treatment regimes, ethically, can those be targeted at young children? And of course, bariatric surgery remained, up until very recently, the only treatment for your morbidly obese diabetic patients. Successful treatment was bariatric surgery. And of course, a lot of the information that we get on bariatric surgery came from us from the States, who very, very early on adapted Roux-en-Y bypass surgery as their main treatment of obesity and diabetes. And of course, the success of bariatric surgery was attributed in part to the altered physiology of gut hormones. And we'll talk a little bit about that going forward now. So again, this audience, all of you know what hormones are, but importantly is that hormones can shape and guide human behavior and physiology. And when it comes to the emotional attachments that we form to food and the social environment that we consume food, it's almost impossible to extract the behavior and the physiology when we're looking at dealing with an obese patient. It's important to also remember that the pathways that regulate energy homeostasis is controlled by only a handful of hormones. And the pathways that regulate hunger and satiety are very ancient and they're complex, and many have alternatives. And an additional complexity is that the hormones and their receptors often have multiple and overlapping functions. Anybody here knows that if you knock out a gut hormone in a mouse, whether it's CCK, GLP-1, GIP, oxytocin, ghrelin, all of these mice are absolutely fine. There are no phenotype. You can force a phenotype to the surface, of course, by challenging these mice with a high-fat diet. But under normal circumstances, most mammals are fine if they lose one or two gut hormones or one or two of these receptors. So the flexibility of our metabolism is extraordinary. And we evade losing, we actively defend our fat stores very, very successfully. So this is, I want you to focus on this sort of day 2013. So this was one of the first sort of major reviews that was published on all of the information that were coming our way on Roux-en-Y bypass. So Roux-en-Y bypass mainly involved a complete dissection of the stomach. And the only thing that remains is a little sort of an egg-sized pouch, about 50 milliliters of the stomach that remains. And then, of course, the stomach itself is rerouted to a distal part of the small intestine, so you can still have bile and pancreatic enzymes can still enter the digestive system. And what they saw with these patients is that the diabetic patients would report this metabolic U-turn within 48 hours post-surgery. So these patients would suddenly have an emergence of insulin sensitivity way before any weight was lost, that you have this metabolic rewiring in the absence of weight loss. And this was contributed initially that this sort of the abnormally high exposure of the distal small intestine to food would lead to this exaggerated release of GLP-1 and PYY. And this led to this dramatic stimulation of insulin secretion. And there was a lot of discussion surrounding the other changes in appetite. And these patients, so usually they lose weight and they keep the weight off. There's now been follow-up studies that show that up to 10 and 12 years later, these patients remain skinny. So they do not pick up the weight. And all of the patients, 100% of the patients see an improvement in diabetes, and more than 87% of them see complete remission of diabetes. And this happens within months after the surgery. So patients report eating behavioral changes. They lose the drive to overeat. They lose cravings for sweet tasting food, and they report the sudden preferences for bitter or salty tasting food. And they also specifically state that they stop obsessing about food. Patients also develop osteoporosis, and of course, this is because of the weight bearing on the bones that is lost, and also leptin is very important for bone homeostasis. But very interestingly, as a certain population of these patients have an increased susceptibility to develop a dependence on alcohol. And this is something that's now really receiving a lot of attention. And all of this sort of taken together would imply that there's something, the reward systems has changed, that the simple anatomical rearrangement of the gut somehow impacts on food-seeking behavior and the reward systems of the brain, which was entirely unexpected. So it's not just a metabolic consequence, but also seems to be a higher brain consequence. And then there's a general idea that obesity is a reward deficiency state with a resultant compensatory overeating. And it's very difficult to, it's important to make a difference between need and want in the brain when we're actually hungry or when we crave something sweet. And there seems to be the idea that's emerging in the literature now is that you've got sensitized wanting centers leading to increased motivation to acquire food. And this is coupled to a blunted reward responses to palatable food. So, and this is well supported. We know that patients with a higher BMI seem to have lower dopamine D2 receptor availability. So there's a lot of evidence surrounding these notions. And I just want to very, very briefly introduce you to the appetite sensing centers of the brain. I'll make this very quick, but it's important you sort of know what I'm referring to. So the appetite regulating circuits in the brain are divided into your hunger circuit and your satiety circuit. Hunger circuits are neurons that project from the arcuate nucleus to the paraventricular nucleus, and they typically express the neuropeptides MPY, agouti-related peptide, and GABA, and they antagonize the activation of melanocortin-4 receptors. On the other side, your satiety circuit, that's driven by leptin and insulin and satiety hormones, are expressed POMC and CART neuropeptides. And these peptides are converted into alpha MSH that activate melanocortin-4 receptors. So you've got this balance between activation of your receptors when you are full and inhibition of those receptors, or antagonizing those receptors when you are hungry. And I have this picture up specifically, mostly for my FHS students, to point out that the majority of research are done on mice. And of course, mouse brains don't quite look the same as human brains. And we have to remember when we interpret these results that we are looking at two different species. But again, so these sort of appetite regulating circuits are found in the hypothalamus. But what's important, of course, is these other two regions, the VTA. So the VTA is known for the regulation of reward consumption and learning, memory, and addiction behaviors. Is often associated with the activation of the VTA. And of course, most of you will be aware of the NTS, where the vagus nerve from your gut innervates your brain. So the part of your gut that's directly in contact with your brain is through the NTS. And this whole notion that reward systems are affected was beautifully demonstrated by this paper published in JCI by Kimberly Steele. And this was the first paper that directly showed that this anatomical rearrangement of your gut had an impact on the activity of your reward centers. So they essentially fed patients milkshakes that were more or less sweet or more or less fat before surgery. And they made two extremely important conclusions. Patients that preferred sweetened mixtures before surgery had the greatest weight loss. And most importantly, those that had the lower taste-induced activation of their reward centers and that had the highest taste-induced activation after surgery lost the most weight. And what they concluded, therefore, is that Roux-en-Y bypass reset the neural processing of reward stimuli. And this paper unequivocally showed that reward is tightly associated with changes that these patients perceive before and after surgery. And of course, if we look at this sort of summary slide, we know that there's a large number of circuits that impact on our feeding, on our desire to eat, on our hunger, our general energy homeostasis, and of course, the short-term control that comes from our gut, our day-to-day eating of meals, and the long-term control that come from our leptin and insulin stores. And of course, the major challenge in the future is to understand how all of these different systems synchronize to regulate appetite and metabolism in every individual. So if I can quickly cover the gut hormones, so we're looking at GLP-1 and GIP, and this is a very old-fashioned picture now, where you classically saw this one hormone per one cell, so GIP secreted from K cells and GLP-1 from L cells. And of course, they're famous for enhanced insulin release from pancreatic beta cells through G-protein coupled receptor signaling. And they're generally known to decrease appetite, increase peripheral insulin sensitivity, and maintain gut health. And this is your classic fluorescence picture showing the broad basolateral hormone secreting side facing the vasculature, and then the narrow apical nutrient sensing side facing the inside of the gut. But again, it was around about 2013 when the pictures and the literature and the reviews started to change. And suddenly you could see there was a very strong dogma shift away from a lot of the information that was available before then. You can see the shape of the cells are now changing when it became clear that these cells would often interact with neighbors, and they would send out these little feet to interact with the nerves and the neighbors around them. You can see that these cells would express many hormones like PYY, oxytocin, GLP-1, and although the GLP-1 pathway to insulin release in the pancreas is shown in this slide, you can see that a lot of other functions are now coming to the fore, and this multi-system impact of gut hormones is starting to emerge in the literature now. So I'm going to take you through a couple of my favorite papers that were published from 2013 onwards. So I think these are my top 10 desert island papers. If I'm ever stuck on a plane or a train, these are the papers I would love to have in my bag. Because every time you read them, you do find something else that you've missed before. So the first paper was, of course, published by the Little lab. So it's Roger Little's lab in Duke University, the very gifted postdoc, Diego Borges. And I still remember reading this paper, opening it up, and you think, oh my gosh, you just know that nothing is going to be the same after this paper. And what Diego did at this time, he spent a happy 3 years constructing three-dimensional structures of the gut. So this was made possible by serial block phase scanning electron microscopy that just became available at this time. And Diego was able to show that the enteric glia basically penetrates the epithelia, which was not known at this time, and that it made a direct physical contact with gut endocrine cells. So up until this point, it was always seen as an endocrine system where hormones would be thrown into the circulation to travel to a distant target, and was never perceived to be a physical contact with the gut endocrine cell. But from years, Diego then published, and Roger Little published a JCI paper very shortly after, showing that not only was there a physical contact, but the contact was functional. And again, the techniques that were available at this time it was just perfect timing. It could not have happened at a different time. And there was also one very fortuitous aspect of this research, where you're just lucky. So many of you will know that gut endocrine cells are very similar to neurons. And of course, they express completely fortuitously this protein called NCAM. So it's neural cellular attachment something. I always forget the name. But of course, this is the protein that rabies virus uses to infect. You need this protein for a rabies virus to bind. And completely fortuitously, gut endocrine cells express NCAM. And if you see over here, when these little mice were infected with a green fluorescent rabies virus, their gut endocrine cells were infected very specifically by this virus. And they could therefore detect these gut endocrine cells. And next, of course, they engineered a mouse, And a lot of these advances were driven by the availability of the correct mouse models. And of course, RABG is a receptor needed by a synapse for rabies virus to jump across. And when they made their RABG expressing mice that had RABG expressing only in the intraendocrine cells of the gut, you could see that following an enema, this infection was carried across the synapse, and you could see downstream infection of the neurons. So there wasn't only a physical synapse, this was an active working synapse. And the next step, and you can see how the impact factor of the papers just increased, that they showed in Science, because the paper showed in Science, that you essentially have an uninterrupted circuit between the gut and the brain. And you could see if you had infection of your gut with the rabies virus, you could see infection travel all the way to the nodus ganglion of the brain up until the VTA and the NTS of the brain, where we know that the vagus nerve interacts with the brain. And they then took this a step further by actually doing electric physiological recordings, so they could directly record action potentials within the vagus nerve. And they could say that the arrival of nutrients, so when you had intestinal perfusion of nutrients in the gut, that the depolarization events that take place in the gut endocrine cells was transferred to the vagus nerve, that you had a single electrical circuit that could run from the gut to the brain. And this was truly extraordinary. We look at the firing rate that was measured in the nodus ganglia in response to sugars, sugar analogs, and sweetness. And what they did in this paper specifically, there was a long-standing argument about what is the difference between ingesting sugar and ingesting artificial sweetness. How do these interact with our physiology? So of course, on our tongue, they taste sweet, but what happens downstream? Is there an incretin effect? Does it make us feel full? And through some very elegant experiments, and again, I just want to highlight this beautiful negative experiment. So they used a technique called optogenetics. And with optogenetics, you express a wavelength sensitive channel. And in this case, they used a chloride channel. And if it's illuminated with a specific wavelength, these channels would open. And of course, when you have an open chloride channel, the cell becomes hyperpolarized and silenced. So this is a technique to specifically silence a cell of your choice. And what they show beautifully, that when they silence the gut endocrine cells, that the recordings of membrane potential firing in the vagus nerve could be taken away or switched off. So again, beautiful negative control to show that when these cells are silenced, the electric activity in response to different sugars then cease. And they use this technique to then further classify the type of receptors that are binding and responding to artificial sweetener and to sucrose. So for example, we know the sodium-dependent glucose cotransporter, and these are the ones that depolarize the membranes and fire your action potential. On the other hand, of course, sweet tasting receptors, by the time they get to your small intestine, they can fire action potentials through G protein coupled receptor signaling. And by exploiting these two different signal transduction pathways, he could then tease apart to show that, that sugar is signaled to the brain through glutamatergic signaling, while of course sweetener a signal to the brain through purinergic signaling mechanisms. And this is a very elegant mechanism to finally show why we are rewarded for eating sugar. Your brain glutamatergic signaling, you are rewarded for consuming sugar, while eating artificial sweetener does not make you feel good, and it does not make you feel full. So your brain can tell when you're eating something worthless, that is, in your brain's opinion, not worth eating. There's no calories associated with this, and you are not rewarded for eating something that contains artificial sweetener. It's a very lovely, lovely paper. But at the same time, there's a different lab, Zachary Knight, that's based in San Francisco, that was very interested in food selection and food and food choices. Why do we choose what we eat? Why do we dislike certain types of food? And he had a very interesting approach to looking at the way that can we tell the difference between nutrition and what makes us avoid toxic food. And again, the development of the technical methods that allowed us to actually investigate these systems. So many of you may know that gut endocrine cells are few and far between. There's very few of them around. They're very difficult to get hold of. And with the start of single-cell RNA sequencing, Zachary's lab then collected all of the gut endocrine cells. I think they collected almost 7,000 of these cells that they all pulled, and they sequenced each one of them individually. And when they looked at this data, they could see that endocrine cells sorted into 8 clusters. And this finally led one of the points of contention to rest. So this one cell, one hormone a notion was no longer valid. What we now know is there's about 8 clusters of different cells, and they all express a wide profile or a different variety of gut hormones. And this expression profile can be altered by the content of your gut. So if you're high fat, high sugar, the expression of your gut hormones can be altered. They then developed, they helped us cross a major technical hurdle and the availability of good mouse models. So inducible tissue-specific markers appear rapidly in organs with a high turnover rate. So if you do a tamoxifen-inducible green fluorescent protein in the mouse gut, it is lost within 48 hours. It's essentially useless for all practical purposes. And of course, a lot of the pre-pro-hormone peptides are expressed in different cells. So pre-pro-glucagon is expressed in alpha cells as glucagon, and it's processed in GLP-1 and gut endocrine cells. So if you have a Cree mouse model for pre-pro-glucagon, are you witnessing the effect on the pancreas or actually the effect on the gut? It was impossible to dissect the various physiological impact of these hormones. And of course, what you can see down here, the Cree models, mouse models, also label sensory and motor neurons. And what Zachary's lab did is they developed a highly specific intestinal epithelial mouse marker. They then had their marker Cree mouse that they would cross with their flipping, with their Villi markers, and you could see that they had highly specific labeling. For the first time in the field, highly specific labeling of our different gut endocrine cells. So you could now see GLP-1, GIP, CCK, oxytocin producing cells highly specifically, which wasn't possible previously. And to make a long story short, he also then used DREADs. So DREAD is designer receptor exclusively activated by designer drugs, G protein receptors. And what it means is these are physiologically inactive G protein coupled receptors. They only respond to a designer drug that you can inject into your mice, or you can add it into their drinking water. And you can selectively switch on G protein coupled receptor pathways in your cell of interest. And he then used the Cree marker mice that he generated in the previous figure to generate these DREAD Cree marker mice so that you could have highly specific activation of, or you could activate the release of hormone of your choice. And he showed that, so he had 2 experiments, one with sucrose that has nutrition, but then ones with flavor lacking nutrition. And he could show that the co-administration of this molecule along with a sucrose meal would lead to strong aversion in these mice for sucrose. And that this aversion was TAC1 positive. So it was serotonin, it's driven by serotonin. If you did the reverse, when mice were trained to choose its flavor that they liked, and when they would co-consume CNO with the flavor, you could see that they would be massively prone towards to select this flavor. And this was CCK and GLP-1 driven. So this was the first paper to convincingly show that gut endocrine cells affect food-seeking behavior and drive preferences and aversion. And of course, this dovetailed beautifully with this paper that I showed with the Roux-en-Y bypass patients that showed that report changes in taste and food preferences and how this links up with activation of your reward systems. And of course, and all of this comes down to the development of our GLP-1 receptor agonists. So this is the new wonder drug on the market that has received a huge amount of media attention. So I think the GLP-1 receptor agonist is sold under the brand name Wegovy in the UK. And there is absolutely no doubt that it is highly effective in treating a morbidly obese diabetic patient. Patients lose a large amount of weight, and they see a very good remission in diabetes on patients that are taking these drugs. There is, of course, further development of this triple GIP, GLP-1 glucagon co-agonists that are now currently in clinical trials. And these are the drugs that for the first time really compare to bariatric surgery in the amount of weight that these patients lose and the amount of weight that's kept off in these patients. But large amount of side effects associated with these drugs. Of course, crippling nausea, the very common side effects of these drugs, as you would expect with a lot of these receptors that are expressed on the vagus nerve. And these receptors, the GLP-1 receptors, are expressed in large and very varied organs in the body. And you can see that out of an endocrinology point of view, you know, I feel a little bit uneasy when you look at the slide and you think we're targeting all of these systems in tandem, you know, and all of these systems are involved in feedback loops. So I want to very briefly touch on two clinical trials that was run. So this one was published in the New England Journal of Medicine in 2021. This was a study run by Novo Nordisk, where they looked at liraglutide, which is a GLP-1 receptor agonist. And of course, patients were put on this 800 calorie per week diet for a couple of weeks, for 8 weeks, and they lost a large amount of weight. And then you had reintroduction of food. And you can see that patients that were on a placebo didn't lose weight. And when they compared liraglutide and exercise, you can see that exercise was as effective as the drug in helping these patients lose weight. And of course, you've got a nice additive effect if you do both. But what I want to draw your attention to is this general health perception. And you can see that patients that take the placebo or liraglutide, so you looked at the blue and the gray line, they felt, you know, equally low, or their expression of health was the same. While the patients that actually did exercise without or with the drug perceived themselves to be feeling a lot better. And I think this is the one thing I want to highlight, that the mental health benefits from exercise should never be underestimated when we are looking to treat a morbidly obese diabetic patient or just an overweight patient. The advantages from exercise alone, no drug is needed in this case, just some good medical supervision and access to exercise. Another one that was published in the Lancet more or less at the same time, and this was just an academic group that almost published it as a response to the Novo Nordisk study, showed that, again, same regime, patients were given 800 calories per month, for 4 weeks, per day for 4 weeks, and patients lost weight. And the weight was kept off with medical supervision and graded introduction of food. So very important to realize that, of course, we can treat obesity in the absence of drugs. But having said that, it's now very well established that the further away you live from a park or a leisure center, the least chance you have of going to swimming or spend some time outside, the more likely you are to be overweight and depressed. And then getting medical treatment to patients that live very far away from medical centers and medical spaces, that can't readily have access to a place where they can exercise safely. It's very difficult to treat these patients. So of course, the logistical aspects of running a study like this is, you know, that's a reality. We can't get this sort of treatment to a very large number of patients that live in isolated places. But if you can quickly have a look at the impact of epigenetics. So there was a lot of talk about, is obesity heritable? What is the impact that the metabolic state of the mother or the father has on their children? What are the genetic implications or ramifications of this? And for me, one of the most important studies that came out was a study by Donkin et al. That looked at the methylation or the DNA modification of the sperm of men before and after bariatric surgery. And a large number of genes that are associated with metabolic regulation did come up. But most importantly, you may remember melanocortin-4 receptor, which is the main receptor that regulates your perception of satiety and hunger was one of the main genes that were differentially methylated and therefore differentially expressed in these men. Of course, the one question that remains with epigenetics, of course, is penetrance. Is this really inherited? So, of course, they didn't check if any of these men happened to have children before or after bariatric surgery, and then went to look at the appetite of these children. Of course not. But I think there's a compelling argument to be made that the metabolic state of the parent is heritable. These changes can be transferred to the gametes that we now know. And one of the most thought-provoking papers for me that came out a couple of months ago was this paper done arguably on worms. So it is on C. elegans, but that comes with its own caveats. But this was the first paper to show directly link food deprivation to heritable behaviors. And this paper for me had two very compelling aspects. So what you could see, what the scientists did is they hatched the eggs and then had starved these worms in the presence of a very specific odor, and after which they would be allowed to recover without any odor. And as you can see on this side, so you can see that, of course, after having been trained, these worms would, so these are chemotaxic studies, so the percentage of worms that would avoid a certain area. And you can see over here, the worms that were trained that a certain odor was associated with hunger will move, will not go into areas where that odor is present. So they remembered how starvation smelled like, essentially. And this knowledge was heritable to the F1 generation. So worms that had these ones as parents also avoided areas that smelled of, that had that particular odor. So they remember, there was some associative memory that triggered, that had starvation to that particular odor. What I find almost more interesting is on this side of the curve, is the fact that avoidance behavior could not be easily unlearned. So if you have these worms that you're exposed to starvation in the presence of the odor, if you then expose them to food and odor, it took 3 tries before the worms forgot that that odor was associated with starvation. But what is fascinating, as the F1 generation, memory extinction wasn't possible. And with the F1 generation, so these ones that were born with the memory, with this associative memory of starvation and odor, it doesn't matter how many times you would reintroduce food along with the odor, they could never change their fear or their avoidance of this odor. And I think this makes for very powerful, of course, these are worms, but we have to think that if food preferences of parents is truly heritable to children at a genetic level, this really has very important implications for how we look at in dealing with obesity in children. It's always been looked as a nurture problem, but it could really be a combination of nature and nurture when we're looking at dealing with diets in children. So last but not least, just to put a little extra cherry on the top, of course, 50% of what's going on in obesity, we still don't really understand. And I love this unexplained phenotypic variation. And just to very quickly show you this these mice are genetically identical, while this one had a UPV in insulin signaling pathway. So there's still a lot of the dynamic regulation of genes that impacts on metabolism as well. But I just want to conclude the talk with briefly looking at, again, coming back to the social aspect of eating. So there's been a lot of pushback against these anti-obesity drugs. And of course, Jay Reiner that wrote, "I've never been thin, but if these anti-obesity drugs put me off my dinner, count me out." And of course, it was a very passionate piece written by Leila Latif that I found very touching, because she speaks with great emotion, you know, that food has always been a fundamental to my identity. My mother was a brilliant cook, taught me how to bake. She fell in love with her husband and themed evenings that they paired with movies, with food. She speaks with great love about her grandmother and memories of her father and the way that they used to cook together. And of course, one thing that we all know intuitively, you understand the country when you eat their food, this connection between a cultural identity and the consumption of their food. And as a scientist, I found this last bit very interesting, where she said, you know, I still cook, but it's a mercenary task. And basically, she's discovered that I can still take pleasure in eating things connected to treasured memories. So she can still take enjoyment from eating food by coupling that to a pleasant memory. And I think we have to remember that we form very strong emotional bonds with the food we eat when we are surrounded with people we love and spaces where we feel safe and valued. And these connections, if they are disrupted by taking some of these drugs, you have to think about the mental health implications of these drugs in the long run. And of course, I am looking at the online environment that so many of our young people and our patients may interact with. This is something that I found fascinating during COVID. And this, of course, is the big popularity of a thing called mukbang. So this is South Korean, was Korean. That means an eating show. And this is where the host consumes various quantities of food while interacting with the audience. And I mean, and it's really the subscription and the viewing of these mukbang broadcasts became hugely popular during COVID. And this is sort of believed to be a result of increased feelings of isolation and loneliness, and as a loss of culture of eating together as social groups. And what you can see here is a YouTube broadcast by a South Korean actor called Seo In-guk. And he is having a traditional South Korean meal. But what's important is to have this is a microphone. And it's not just the eating of the food and him describing the food, but it's the sounds of him crunching into fresh lettuce and slurping the hot noodles. So it's not just seeing somebody eat, but listening to this person. It's a very personal broadcast, and it's very, very popular, very interesting. And of course, last but not least, the one thing you can't have this talk without is to point to our very personal link of our perception of beauty to our body weight. Our body weight through the ages, whether you're fat or thin, people of different weight has been considered to be beautiful in the past. If you do have time, please go and look at the Welcome Collections exhibition that's up until April now in the pursuit of beauty, perception of beauty through the ages. What I love about this, they poster on their website, as you can see, these two models are most certainly considered to be beautiful today. BMI well below 25, right? Two very skinny young people right here. But if you look at the back, at these three very plump women in the back here, these, I mean, of course, were considered to be very beautiful 500 years ago. And of course, today, no doubt, they will be on their way to their GP to go and ask for GLP-1 receptor agonists. So I think our connection with being skinny and beautiful, we have to recognize that a large number of people that are taking these drugs are using them that are not actually overweight. And there's now a huge population of people, especially, Hollywood, they refer to the skinny jab as Hollywood's worst kept secret. So a large number of people consume these drugs, not because they're overweight, but because they want to be very skinny. And of course, we haven't touched today at all on the other side of the coin when you're looking at eating disorders, such as anorexia. So in conclusion, as John Severin said many hundreds of years ago, tell me what you eat and I will tell you what you are. He was, of course, as far as I'm concerned, the first really mukbang artist, because he wrote elaborately on food, the Physiology of Taste, where he celebrated both the cooking and the eating and the culinary reminiscence surrounding food. So, of course, we have a very complicated relationship with food, and in step with this, we have an intricate network of electrical excitable cells that is sensitive to ingested food. And this circuit runs from the gut to the brain. Our electrical circuit converts food into signals which coordinate gut motility, secretion of hormones, food intake, food preferences, metabolism, mood, and behavior. So all of these aspects of our eating behavior is affected. Enjoying food is universal and it transcends cultural, religious, and social boundaries. And we need to understand what drives the mechanisms which make food appealing, and why do we crave different tastes and different colors and different textures. And all of this will help us move forward if we want to have effective intervention of our obese patients. Thank you. What I would like to add, anybody that would like the PDFs of my talk, please just send me an email. I would be happy to send you a copy of the talk that I